Hi everyone, thank you for being here. Thanks for the invitation. So as Maggie said, I'm working in a constitutional cytogenetics disease department. So I'm not gonna talk about cancer, but I have a lot of examples about developmental and reproductive diseases. So this is my disclosures, no conflict of interest to be declared. I'd like to start by few slides just to have an overview back on how chromosomes looked before and how they improved. So as you know, we started with Fleming's drawings of chromosomes, and with the development of cell culture and bending, we get these nice chromosomes today, but still they are not perfect. At least they deliver not enough information for us. And then starting from the 80s, you had this revolution of molecular cytogenetics with hybridization and comparative genomic hybridization and DNA microarray as well. And this led to a real improvement of the diagnosis yield. Well, so now we have actually this very nice diagnosis yield with the CMA, chromosome microarray analysis, but this is for CNV detection. And we are still struggling and still challenged by for the detection of balanced translocations or balanced inversions, etc., because we have only karyotype that is able today to do it in a real clinical setting. So you are here at the ESHG. You hear a lot about sequencing. Yes, we had a sequencing revolution this last decade, and it's getting cheap, fast, but SV detection is still a challenge even for sequencing. So to my knowledge, up to now, there is no study that has shown that NGS could eventually replace CMA or karyotype in a diagnosis setting. And as said before, this is limited because we have a lot of short read sequencing that is commonly used, and we have these low copy repeats at the ESV breakpoint, so this is a hurdle that is really hard to overcome with sequencing. That's why current guidelines, they recommend to use CMA as a first-line test for developmental diseases. And for reproductive diseases, you have karyotype that is recommended because you have up to 15% of chromosomal abnormalities according to the type of infertility. And in these chromosomal abnormalities, you have a lot of aneuploidies and structural variations that are balanced. So how we get involved into this technology? Well, we've been really excited, as Alexander has been, because I think we see the same chromosomes at the end in this technology. And we aimed, our study at least aimed to compare Bionano-optical mapping to regular pangenomic analysis tools that we have in a cytogenetic labs, that is karyotype and microarray, but also some, for now, research tools that is whole genome sequencing. So we are aiming to compare their sensitivity and also accuracy in defining breakpoint location. So this has been a multi-center French study that involved 3 main collaborators, me at the Cochin Hospital in Paris, our collaborators in Lyon and Dijon. And we have included for now 30 patients that have been seen in our genetic center, either for reproductive diseases, mainly sperm defects and miscarriage, recurrent miscarriage, of course, but also developmental diseases, autism spectrum disorder, and intellectual deficiency mainly. So there was a subset of these developmental disorder patients that has also been included in another study that is called the YANI project, whatever. The important thing to know is that in this project, these patients carried de novo apparently balanced structural variants, and they had, of course, karyotype without or with microarray analysis, And they got also a whole genome sequencing by short reads. So in this study, we included a wide variety of abnormalities. One case of an employee, to answer to your question, it might, so now I don't think I have been silly including this case since other people asked the question. And we had also some unbalanced as well as apparently balanced structural variants, roughly half and half, deletions, duplications that span from 200 kb to 5 megabases. So really we wanted to have some kind of a whole view of what you could expect in these situations. And these are the results, at least the QC metric results. So just on the left to explain how this works. So first your samples after extraction, you had the clinical details, but you will de novo assemble and compare the de novo assembly to a reference genome. This using a structural variant calls algorithm will provide you a list of variants, as you see here, the means. So these are several hundreds, thousands of variants, and these will be filtered out, as you heard, by using a filter that we chose at 100 kb with Bionano, which makes sense regarding the pathogenicity of the CNVs. And you get a final set of SVs that is reasonably comparable to regular techniques. So in addition to this, you get also CNV calls by the CN algorithm or coverage, as Alexander showed you before, and we will see some examples. So this is an overview of the results. You have just to look at the blue and green columns. So because we are still analyzing some of them, that's why. So among the analyzed samples, you can see that we almost recovered almost all the cases, and the ones that we didn't, I will show you the explanation. So let's go over some examples. So first one, a male who comes for azoospermia. His karyotype shows a very common one in this case, compatible with the Klinefelter syndrome. And here you have this circus plot, and you have just to look at the coverage here. You get, so 23 is the X chromosome, 24 is the Y chromosome, and if you compare the coverage of X chromosome to the autosomes, you can see that there are 2 copies of X chromosome, and for the Y, you have half, so 1 copy. And just to compare to normal karyotypes, so this is a female, 2X and no Y, and same for a male, you get this coverage, 1X and 1Y. So fortunately, it detects. So second case, a female with endometriosis. She got karyotype and it shows an inversion. So it's a pericentromeric inversion of chromosome 3. The inverted one is right here. It's quite a big inversion, so it was visible on the karyotype. And these are our results with Bionano. So on the left, the circus plot that shows something going on chromosome 3. And here you have a zoom out on this region. So this is the reference chromosome 3. And you have these 2 maps of our patients that map on either sides, which correspond to fusion points of the inversion. So you get exactly your breakpoints right here and here. So these breakpoints match with what we thought were the breakpoints based on the karyotype, and it allowed also to correct our breakpoint location. Third example, a male with oligoastenoteratozoospermia. This is also an indication for karyotype. And here we had a translocation between chromosome 7 and 21. So as shown for those who like karyotypes. So this is the derivative chromosome 7, that doesn't look like the normal P of chromosome 7. And this is the derivative of chromosome 21 that has the P of chromosome 7. So these are the results from Bionano. And as explained before, so the blue map is the patient's map. Above you have the reference chromosome 7 map. Below, reference chromosome 21. And as you can see here, you get perfect match between your patient and this part of chromosome 21, 21q. And then you have a perfect match between your patient and the map of chromosome 7. So this corresponds to the fusion point of the derivative chromosome 7. And you can have the same here on the other derivative. This is the derivative chromosome 21. Fourth case for reproductive disorders, and last one for these indications. So this is a male, again, azoospermia karyotype, showing, again, a classical anomaly that you can find with azoospermia. This is a mosaic, 45X, 46X with isodiacentric chromosome Y. And you can see by G and R banding that it can be tricky, but well, we noticed it. It's not always easy, that's why these technologies could be helpful. This is a result for fish to confirm. So this is a YP telomere with some backfish on the YQ. This is the result from Bionano. So you have a whole view of chromosome Y. This is a normal male, normal result of Bionano for Y chromosome. So you have this all this region that is missing corresponding to the heterochromatin of chromosome Y. This is our sample, a patient, that has an isodiacentric chromosome with a breakpoint roughly at this level. So you can notice that you have no coverage of these regions, which support a deletion of this whole region, okay, compatible with isochromosome Y. But we did not notice anything wrong on this side, which makes sense because we had a mosaic between cells that do not have at all chromosome Y and cells that do have 2 copies of this region. So since the mosaicism was half and half, roughly, that makes sense, okay? So just to say that this will be anyway a common problem, not really, because anyway Bionano detects it, whereas NGS cannot detect even this deletion. So the important point is that you can suspect something going on wrong on chromosome Y, then you can check what's going wrong by karyotype. So then I have few examples on developmental disorders. So I didn't want to be too long. For microdeletions, microduplications, we will not see them since Alexander showed some, but they have been all detected. I'll show you just 2 very unusual examples. First one, male with developmental disease. So CMA shows this profile. This is chromosome 8, the P arm of chromosome 8. It shows a deletion of the terminal part, then a normal part of chromosome 8, and followed by a duplication of this large chunk of chromosome 8. So for those who are used, this is something that is compatible with the famous inverted duplication and deletion of chromosome 8 on the P arm. And this is shown also by fish analysis. You see that this is duplicated and inverted. The telomeres are deleted. These are the results from Bionano. And what is nice is that you have the profile that looks like the CMA result, deletion here, duplication after, and here euploid region. And in addition to this copy number algorithm based on coverage, you have this structure algorithm, SV algorithm, that shows you that there is actually an inversion at this point. So you have the answer as FISH and CMA provide you. Second example, male, developmental disease, intellectual deficiency. This is a rare case of chromoanagenesis because CMA and karyotype showed that there were multiple events. So this is part of a chromosome 3. It's the whole chromosome 3, actually. No, part of it. And it shows many regions that are deleted. When we performed the karyotype, we got this multiple three-way translocation, actually, as shown on the karyotype between chromosome 3, 5, and 13. These are Bionano results. On chromosome 3, you can notice that there are multiple events, and in addition to that, you have a translocation event between chromosome 3 and chromosome 5. This is a zoom on these two chromosomes that shows the multiple events on chromosome 3. And this is the map view with all these possible fusion points that match somewhere, everywhere into chromosome 3, which supports the chromoanagenesis, sorry, and this is consistent with NGS results. Last examples are about this cohort that has been also sequenced by Illumina, whole genome sequencing. So this cohort of 55 patients led to detection of 49 out of these 55 abnormalities by whole genome sequencing, and 6 of them were not detected by whole genome sequencing. So we picked up some examples, 9 from here that we run with Bionano, and 4 from these guys. So for now, I have the results for 8 of these, and as you see, these are the different translocations, one inversion, among which the 3 that failed to be located by NGS. And with Bionano, we could locate the breakpoint, detect and locate the breakpoint for 2 more compared to NGS, and the others as well. So among these, I think we don't need to go through into the details, but just to show you one example, this was a translocation between chromosome 7 and chromosome 10. We did not know why exactly NGS did not pick up this translocation, but now that we have the result from Bionano, we can go back to the NGS results and try to understand why it did not pick it up. So I had some, let's say, failed cases, even if we are working hard with Bionano to understand why. So this one was a female who came for reproductive disease, who had an inversion on chromosome 10, pericentromeric inversion. And for now, what we see is that breakpoints potentially locate into regions that are poorly rich in DLE1 sites, the labeling sites of Bionano, so that might explain why we couldn't pick it up. And the second one, it's not really a failed sample, it's just to show you what happens when there is a breakpoint into the pericentromeric region. So this is a derivative chromosome 14 from a translocation between chromosome 11 and chromosome 14. As you can see from the karyotype, a piece of 11q terminal went on top of the chromosome 14, right at the pericentromeric region. So we saw the trisomy, the partial trisomy. Yeah. We see the trisomy, so that's as CMA. The only thing is that we don't have any clue on the translocation on chromosome 14. So then you can go further onto your case and have the explanation. So it resembles a CMA result for this case. So that's how I conclude. For my experience, this is an easy process. We've been really happy with the high success rate. We did not have to redraw samples or re-extract or whatever. We had an efficient identification of structural variants with no overwhelming calls. So Bionano came just after a test with 10x genomics, just to not to mention them, but like there was a huge difference for us. And at least for the isodiacentric chromosome Y, we had this indication with the deletion that has been detected. So at least we could suspect something going wrongly. And we succeeded as well to identify 2 more samples, 2 more breakpoints compared to NGS. So we would like to test the mosaicism level threshold, even though I saw just that you've tested some for somatic diseases. I did not know, because this is something that happens often in reproductive diseases, to have monosomy X in a mosaic state or isodiacentric in a mosaic state, whatever. We know that there are possibilities to improve the technology. I'm sure that the inversion 10, probably we will find out what happens and we can improve the SV call. We would like to evaluate the precision in defining breakpoints, but for now, at least for copy number variants, we were really close to the breakpoints that were found by CMA. So we are quite confident for that. And we are going to dig into the genotype-phenotype correlations, obviously. Last thing to mention, but it has been mentioned, there is a limitation for these centromeric regions, but well, I'm confident that we might find a labeling because, as we already said, there are fish labels for centromeric regions that are specific, so there might be some specific regions for centromeres. So this is a good point, and throughput is improving anyway, so it's a positive element. So just to discuss, so the question now is, since we are able to detect efficiently balanced as well as unbalanced variants, so is Bionano a milestone now in cytogenetic history? And I might be wrong, but I believe that for now it's possibly yes. Could this become the next generation chromosome analysis tool? I think so. Well, I was glad that you put the same figure than me. Yeah, I see chromosomes in here, so we have the same vision of the technology. And the answer to this, yeah, it depends. So to me, at least in developmental diseases, now that we've put CMA as a first-line test, we know that we lose diagnosis for situations with de novo, apparently balanced translocations, inversions, that could potentially be pathologic. We know that it's around 1% of cases. So at least this technology would recover these cases in addition to detect the copy number variations. And for reproductive diseases, this needs really to be evaluated. You have to wait between the increase that you are expecting in yield diagnosis, meaning that you will anyway recover cryptic rearrangements, because honestly, on the case that I showed you, it was really hard. It's one of my colleagues who saw the translocation. This translocation is really hard to detect by eye between this piece and this. Honestly, some cytogeneticists, I think they would have missed this one. So at least for cryptic rearrangements, we think that it will improve the diagnosis yield. But on the other hand, you might lose some diagnosis because you have these rearrangements that will still be for now undetectable, the Robertsonian translocations, but hopefully it will improve. So that's why we need to think about what we are ready to lose, as we did for CMA. We said, well, 1% of de novo translocations, finally, we gain more with CMA than we lose without karyotyping, so why not? So at the moment, I think that there is essentially evidence that we need multiple tools anyway for now, and we need especially to establish algorithms that define exactly first and second line genetic investigation strategies. So with that, I thank all my colleagues at Cochin, my collaborators, different hospitals, the Human Genome Research Center in France, and the Bionano Research Department, because they helped me a lot analyzing the data. They are really available. And as Alexander did, yeah, we know that these products are for research use only. Thank you.